The term complement refers to a complex set of distinct serum proteins present in normal human serum. The term complement refers to the ability of these proteins to complement, supplement, or to augment the different effects of other components of the immune system. They aid in the antimicrobial effects of antibodies. They are also considered to be an important component of the innate immune system. These are a set of complex proteins of approximately 20 in number. They are involved in three separate pathways of activation. These complement components are synthesized in the liver, except for C1. These components are numbered from C1 to C9, and all 9 components consort or interact with one another. Fragments of these components are given the lower case. For example, a fragment of the C3 component is named as C3. A is usually given to the smaller fragment, while B is given to the bigger fragment. The complement system has three main effects which will be discussed in the video. The first one is the lysis of cells or cytolysis. Second one is the generation of different mediators. And the third one is oxidation. The components of the complement system involves more than 25 molecules whose biologic activities are diverse and closely related. 5 proteins are usually seen for the classical pathway. 3 proteins are needed in the alternative pathway, and 6 proteins are common in both pathways. This make up about 14 basic and distinct proteins. So what makes it more than 25? We need to add the seroproteins that modulate complement-derived activities, like the C1 inhibitor, plus we need to add other molecules that participate in the complement system. Like plus, all of this would make more than 25 molecules that participate in the complement system. There are 3 pathways involved in activation of the complement system, and these are the classical pathway, alternative pathway, and the lectin pathway. The activation of the different complement is often referred to as a cascade, which begins when one molecule is activated by certain reactions, and the 3 different pathways all involve the cascade of the different components. This is a table that differentiates the pathways according to their activation and the different requirements that the unique components present each for the activation of classical pathway. Needs in immune complexes or the antigen and antibody complex. The alternative pathway and the lectin pathway are both antibody independent, which means they do not need the presence of the antibody to be activated. For the requirements, the classical pathway needs at least one IgM antibody or 2 IgG antibodies, and we will discuss this later on. The alternative pathway needs microbial surface or the cell walls of the organisms. The bacterial cell walls have the polysaccharides or the lipopolysaccharides that are needed for the activation of the alternative pathway. The alternative pathway is more important the first time that an infection happens, because the antibody required to trigger the classical pathway is not yet present. The lectin pathway, on the other hand, requires the carbohydrates on microbial surface. The different unique components for the pathways are the classical pathway needs C1q, C1r, C1s, C4. And C2, the alternative pathway, has factor D. Factor D and properdin, while the lectin pathway has the MBL, or the mannose binding lectin. This is the cascade that is followed by the classical pathway. It starts with the presence of an antibody which is bound to an antigen. This is the activation of the classical pathway. The first component recognizes the presence of an antigen and an antibody and starts the activation of the different components of C4, C2, and C3. And C5 forms the first membrane attack unit, and this further activates the different components. From C6 to C9, which makes up the membrane attack complex. Let us now discuss the classical pathway in detail. The activation of the classical pathway starts with the presence of an antigen and an antibody complex. First component of the classical pathway is the C1. This is the recognition unit of the classical pathway, which is a macromolecule held by calcium. It is a trimolecular complex with three subunits of C1 called C1, Q, C1R. And C1s, C1 appears as six groups held together on slender shafts that fuse into a common base. It is also considered as a pentamolecule because it consists of a single C1Q2, C1R, and two C1s molecules. Means of the C1 molecule binds to the second heavy chain portion of an IgM or two IgGs. The globular domains are the recognition unit that bind to the Fc region of the immunoglobulins. C1 needs to bind two Fc portions. That is why two IgGs are needed. Since IgM is a pentamer, the presence of one IgM will initiate the cascade. Once the CM complex is attached, this leads to a conformational change in the C1 complex that leads to C1s molecules. If active serine esteresis, this starts the third stage, which is the activation of the C4 component. C1s activates the next component C4 and is cleaved to C4a and C4b. C4a has no further part in the complement sequence. C4b attaches to the microbial surface. It does not attach to the antibody complex. C4b now acts on the binding site of the second activation unit, which is C2 when combined on C4b. C2 becomes a substrate for C1s and is cleaved into C2a and C2b. C2b is very labile and will decay very fast. C2a remains bound to C4b in the presence of magnesium to form C4b2a, known as C3 convertase. C3 convertase is called as such because it splits C3, which is the most abundant complement. It splits C3 into C3a and C3b. C3a is released into the plasma while C3b combines with C4b2a to form C4b2a3b. This is the most significant step in the complement activation series. C5 converts cleave C5 into two subunits C5a, and C5b C5a is released into the body fluids. The generation of C5b starts the formation of MAC or the membrane attack complex. C5b binds with C6 forming C5b6 and this then binds to C7 to form C5b67 complex. 6, C8 and C9 then sequentially bind to this complex. C9 forms a pore-forming molecule containing up to 14 C9 monomers. This results in the formation of the Leydig block. This is the first function of the complement system. Cytolysis is brought about by the entry of water and electrolytes into the cell through the disrupted cell membrane. This cascade happens simultaneously to different parts of the cell and to all antibody-bound cells, including bacteria, tumor cells, and erythrocytes. The second function of the classical pathway is the generation of mediators, C4a, C3a, and C4b, that were produced during the classical pathway activation. These components act on mast cells and basophils for degranulation. These cells will then release mediators like histamine to increase the vascular permeability of the blood vessels. This allows an increased leakage of other complement and other plasma proteins to the site of infection. Among the three components, C5a is the most potent. It is also considered as a chemotaxin or a chemoattractant that leads phagocytes like neutrophils and macrophages to the site of activation. The third function of the classical pathway involves optimization of C3ps. Their presence on the surface of the cells makes phagocytosis better and more efficient. The second pathway is the alternative pathway. The microbial cell wall activates the C3 conversion, leading to the binding and splitting of factor D. C3 convertase is formed when properdin is bound, cleaving more safely and leading to the C5 convertase. This eventually forms the membrane attack complex, which is similar to the classical pathway. The alternative pathway is antibody independent. It does not need the presence of antibodies to activate this pathway. The alternative pathway is more important than the first line of infection by microorganisms because that antibody requires to trigger the classical lipopolysaccharides or endotoxins. Fungal cell walls are viral envelopes, so how does the alternative pathway work? One thing to know more about the protein C3 is that it spontaneously converted into C3A and C3B. It does not need to be activated to be converted. The normal cells, normal healthy cells, can inhibit the effect of C3b. Once C3a attaches to the cell walls of the normal cells. Foreign cells like bacteria, viruses, and fungi do not have the inhibitory protein to deactivate C3b. If not deactivated, this then starts the alternative pathway. The C3b that is attached to the microbial surface then acts as a binding site for factor B. This is the first unique component of the alternative pathway. The second unique component is factor D. It cleaves factor B, which is bound to C3b to form BA and BB. The fragment BA is released while BB stays with C3b and C3BB. The C3 converters of the alternative pathway property, which is the third unique component for this pathway, binds to this complex to stabilize it. The stable C3 convertase then cleaves further C3A into C3A and C3B. C3A is released while the formed C3B binds to the C3 convertase to become C3BB, BB-C3B complex, known as the C5 convertase. This continues to the final phase of the activation of the complement cascade, which is similar to the classical pathway C5 converts cleaves. The third pathway is the lectin pathway, also known as the Mannan-binding lectin. This pathway does not require an antibody for activation, as well activation is brought about by the mannose binding or MAN and binding lectins are proteins that can bind to carbohydrates. This is produced in the liver and is normally present in serum MBL, also known as acute phase protein. Therefore, increasing during inflammation, the lectin pathway requires the activation of MBL, a unique component of the lectin pathway. The Mort Mannan refers to the polymer of the mannose. MBL is somewhat similar to C1 component. In structure and function, C1, as mentioned earlier, binds to the Fc portion of the IgGs or IgM. A lectin is any protein that binds with sugars or carbohydrates like mannose and glucose, which are found on the surface of the microorganisms. MAS stands for MBL-associated certain proteases. The MAS are very similar to C1R and C1S molecules of the classical pathway, in that it cleaves C4 and C2, so that this is how the lectin pathway works. The carbohydrates found on the surfaces of the microorganisms would be recognized by the MBL, and the MBL will attach to them. MBLs are normally found in serum, so when organisms are seen, they would be recognized by the MBL. Once the MBL is attached to the carbohydrates like the mannose or the glucose on the microbial surface, the MAS2 will then act like the C1, where when C4 is attached to it, will cleave into C4A. And C4B then follows the same pathway as the classical pathway. C4A is released, while C4B is bound to the cell surface and attached to C3b, forming the C5 convertase, and follows the same pathway to form the MAC or the membrane attack complex until lysis is formed. To summarize the different actions of the complementary system, first is that increase the vascular permeability done by the component and that have been released to the body fluids. These components also cause the degranulation of mast cells and basophils that also leads to the smooth muscle contraction. C5a acts as a chemotaxin for phagocytes like neutrophils and macrophages, while C3b acts as obstinans for phagocytosis. And lastly, complement system stimulates the bacteria and foreign cells